till 8 o'clock when we officially start. Where are we at? There, okay. Anyway, as always, like I say, check the course announcements. Make sure we're having class today. Today, I did post and say we will be here. I wouldn't normally, since we haven't been here in a while. I went ahead and let you know. Plus, if you saw the announcement, and as you can feel, the heater is a little bit broken. I won't say broken. It's working. It's just not working very well. So it's a little bit chilly in here. So for those of you who were here, you will get a little extra credit for being here in person. Yeah. Anyway, yeah. So make sure you check those before you come in. Any questions about that? All right. Some other old news. Lab 2 is due today. I'm sure you've seen in the announcements. It was we had to switch it up since we couldn't meet for lab. So instead of doing the original lab worksheet that you had to do, which we would have met in person and done a pH experiment, instead of doing that, do the other lab worksheet that I presented to you in the lab feedback and in the course announcements. And it's basically the same lab, except instead of physically doing a pH experiment with acids and bases, you're going to go to some website and do a pH experiment. Either way, I'm still here to help you, right? So if you haven't done it yet and you need help, let me know. Same with pre-lab 3, right? Pre-lab 3 is due today. And again, if you need help, it's not a test. I'm not saying like, do you understand this, right? I'm here to help you. Let me know if you need help. So for pre-lab 3, I'll give you a heads up. Let me think. So we have at least one video and a set of questions. This is the beginning of the pre-lab, if I remember correctly. And then a set of questions to make sure you understood the introduction. And then for the first time, I think this semester, yeah, you're going to keep going, and you're going to provide a bunch of hypotheses. And then once you're done with your hypotheses, then you're done for the pre-lab. So that's it for lab 2 and pre-lab. Any questions about that? All right, 7:58. I'll slow down. But yeah, so again, since we're a little bit before 8, I just want to reiterate this. I'm here to help you. These are my office hours. This is when you're guaranteed to find me. Not when this is not when me saying this is when I work. That's not what I'm saying. I work a lot all day. So if you need me, even if it's outside of those hours, just let me know and I'll glad to help you. This is, again, just to reiterate, office hours are just when you can drop in online or drop into my office and just know I'm going to be there without having to email first and say, hey, can we meet at 2 p.m.? That's all. So again, please let me help you. Since, again, we've got about a minute left, so I'll slow down. I'll only start any new information. So just harp on that for a little bit. Again, I'm here to help. I was reading my course or student evaluations from the other semester, and it honestly just breaks my heart. Some of the people, like one person said something like, he just doesn't want us to succeed or wants us to fail or something like that. I'm like, how could you think that? And I'm trying all semester. I tell them, I'm telling you, I'm here to help. You do not have to do any of this alone. If you make mistakes, correct them. Get your points back. If you need me for feedback for an assignment that's been graded, you get a 5% boost to that grade if you let me talk to you about the assignment. Yeah, so I don't want you to think that I want to see you fail or that I don't want to see you succeed. That's what I'm here for. That's why I allow you to, do extra credit and correct old mistakes. It's a lot more work for me, but hopefully it helps you. Anyway, really quickly, we've got some more people coming in. And it is 8, so now I will take roll one more time. Attendance. Ashley is here. Barrett is here. Wooten is not here. Trahan is here. Dickens is not here. Clark is not here. Inca is here. Isaiah is not here. Copes is not here. Katie is not here. Kaylee is not here. Lucas is here. I already had you. Sorry about that. Marley is not here. Noah is here. Savannah is not here. Mia is not here. Robert Edwards is not here. And Brooks is here. Did I miss anybody? All right. So, sort of new stuff, I guess. Sort of. Again, attendance is mandatory. Participation is graded. If you're in person today, all I require is just to send me an email to say, hey, I was here. But as always, you can do these other things I'm about to say for the people watching the video, and you guys can get extra credit. And you guys who are here today are going to already get extra points just for being in person. But anyway, so again, if you're watching the video, you're going to send me an email that has the attendance words, screenshot every time I say an attendance word, and then just one question about the material covered in today's lecture. And the first word for attendance, so if you're watching the video, you should be screenshotting me pointing at it. It is escape. Because today is National Escape Day. I don't know what that means, but it is National Escape Day. So any questions about attendance and participation points? You have one new person, so just to read, just so that you understand. When I say it's mandatory, what I mean is the university says it's mandatory, and I have to report attendance to the university. And when I say it's graded, you get 100 points out of 1,000 for being here. Yeah, that's it. So if there's no questions about that, the last announcement is technically the exam is next Friday. Usual times, just come here as you have done today. Only difference is you're going to need to bring your computer with your charger. Obviously, you don't want it to die. If you want to, you should bring a pencil, maybe a calculator. Any kind of math is going to be really basic. We've already talked about like the really basic math you're going to have to do. But if you think you're going to need a calculator, bring it. Obviously, you can't use your phone because I don't know if you're looking at a calculator or ChatGPT, right? So, yeah, make sure you have that. It's going to be 50 minutes, right? Because when class is done, we have to leave. So, all that being said, if you want more time to take it, if you just don't feel like you're a good test taker at 8 a.m. or whatever it may be, contact me sometime before then. If we can make arrangements for you to take it with me, either in person or online, you can maybe take it at another time. But you have to plan ahead. So if you just miss Friday and you haven't planned ahead, you're just going to get a 0. But speaking of zeros and any other grade you get, to ease your anxiety a little bit, you're going to be able to retake your exam. So what will happen is you'll get your grade, whatever it is, and then once everybody's done taking it, then I'll open it back up. And when it opens back up, you can take it as many times as you want. Open book, not timed, up until we take exam 2. So that'll be the window to retake it, from the end of exam 1 to the beginning of exam 2. And what I'll do is I'll average your first grade and then your highest retake grade, whatever it may be. So you have nothing to lose. Well, yeah, let me rephrase that. So let's say you had an 80 the first time you took it. For some reason, every time you retake it, you can't pass an 80. I don't know how that would happen, but if that were the case, we would just stick with your original higher 80. So yes, you have nothing to lose. Make sense? So many times people don't take me up on that. I don't understand. And then at the end of the semester, people be like, what can I do about my grade? What? Should have been taking the exam. A lot of points there. So anyway, that's it for reminders. Do you guys have any reminders? Announcements? You see what that noise was? Make sure we're still recording. I've already been kicked off once today. Okay, good. Marley's joined us. So, Marley, since this is just the announcements, I'll let you know that the first word for attendance is escape. Since you're live, all you need to do is send the attendance words. You don't need to do screenshots. Just send the attendance words, send the question after 9 a.m. I guess we're good to start. But again, sorry, one more time before we move forward, I want to reiterate. We are tentatively scheduled to have the exam next Friday. But if we need to, we can push it back, right? So if we haven't finished, obviously if we haven't finished what we need to finish before then, we're going to push it back. Or if we finish it right before, and then you guys all email me and say, can you push it back? Majority rules, and I will push it back if enough of you want me to. So let's move forward. We were talking about acids and bases. I'm going to move up. We already talked about this. Whoa, what's going on here? Excuse me. We had already talked about this. This is a good thing to study really quick, and before we move forward, I'll just remind you some stuff. You don't need to memorize any of this, right? So there's not going to be some question where I don't give you this and I say, what is the pH of household ammonia, right? You don't need to memorize it. It's somewhere between 11 and 12. But again, I might give you something like this, and I might say something like, how much more acidic is grapefruit than urine, right? And then you'll have to be able to say, all right, well, it's 10, 100, 1,000. 1,000 times more acidic than urine. Or 1,000 times less basic than urine. Or 1,000 times greater concentration of H plus ions than urine. Or, and this is the last one, 1,000 fewer OH minus ions than urine. Does that make sense? If not, we'll talk later. I can give you some examples. Maybe I'll come up with an extra credit homework assignment that you don't have to do it. You can have some practice doing that, but you'll get some extra credit for it. But yeah, just think, when you get questions like that, there's so many different ways to describe the relationship between two different pHs. more acidic, less basic. Anyway, this is the last thing if I remember correctly, that we left off on was that name. So yes, let's talk about buffers. Luckily for us, we didn't actually do the lab that we were supposed to do on Wednesday. Otherwise, this would have been important information to have before we went into the lab. But a buffer, as you're going to need to know for the exam, is a substance that minimizes changes in pH. It's that simple. It minimizes the change in pH. Which makes sense. I should have talked about this when I had this. And I'll come back to that side, don't worry. So think about it. Your pH of your blood is 7.4. For extra credit, if you want to look it up, how much is too high, how much is too low, it's a very narrow window. You will die if it's too high or too low. For extra credit, you can send me a separate email, not this one, a separate email, include your source, tell me how much is too high, too much is too low. But anyway, it's very narrow. If it moves too high or too low, you will die. But think about it. You can drink, I have moved drinking black coffee right now, and I'm hoping I'm not gonna die today in front of you. But I drank black coffee yesterday and didn't die, right? So somehow I drank something that acidic and didn't die. Over the weekend, I definitely had some soft drinks. That's a pH of 3, really acidic. I didn't die. Didn't have lemon juice, I had some lime juice over the weekend. Had a little margarita action. But again, I didn't die, right? That's a pH of 2. Battery acid, I drank a bunch of that. No, I didn't do that. But anyway, the point is, right, here we have all this acidic stuff. Then why don't we die? Why doesn't that kill us? And the answer is buffers, right? So that's what buffers are. That's why they're important, even though I'm probably not going to ask the importance in the exam. You just need to know what they are and how they work. So remembering that they resist changes in pH, that's probably pretty easy, right? It's kind of in the name, it's a buffer. The hard part, maybe, the relatively hard part is remembering how they work. And just remember they just do the opposite, right? So if you put a bunch of acid in there, just remember what an acid is according to this course. A bunch of H+ ions, right? So put acid in there, it's going to do the opposite, which is absorb those H+ ions. When you put in a base, it's going to do the opposite. It's going to give away those H+ ions, right? It's going to try to keep that pH neutral. And then, again, that is exactly what your body does. And again, for extra credit, suggest maybe you can look up some specific examples. Like, what are the buffers? Like, what's the chemical names of the buffers that are in our body? Anyway, are there any questions about acids, bases, or pH before we completely get out of the middle, the second bullet point, and move on to the third and final portion of this chapter? All right, you guys nice. Don't be afraid to ask questions, especially considering it's been so long since we've seen each other. You might have a question you think's dumb, but your classmates might be thinking, I wish I could ask this, but I'm afraid to ask this. So if you have questions, ask. Anyway, I know that's really hard to see. But again, this is the third and final portion of the chapter, and it is biological molecules. This is what we're going to be having a lab about next week. So this is what your pre-lab is about. And for those of you who've done the pre-lab already, good on you. Like, it's great, right? Doing it early is the way to go. But in this case, it almost stinks because now you're going to have a little bit more of an insight of what the biological molecules are. So your hypotheses would have been more informed. Not that it matters, right? Because you're not going to get graded if your hypotheses are correct or not. Anyway, I know you can't read this, so I'll say it very quickly for you. Here's the learning objectives. By the end of this, you should be able to describe the ways in which carbon is critical to life, which probably won't necessarily be a test question, but that's okay. You should be able to explain the impact of slight change in amino acids on organisms. That is true. You should know that, and we'll really get into that later in the semester. And I haven't told you what an amino acid is yet, so if you're wondering what's he talking about, don't worry about it. We'll get there. You should be able to describe the 4 major types of biological molecules. That's the biggest one, I think, as far as the exam is concerned. And finally, you should understand the functions of the 4 major types of molecules. So let's jump into it. As you already know, cells are mostly water. Just out of curiosity, I don't expect anybody to have memorized yet, but does anybody remember the number I gave you? For how much, like, your cells, like what percent your cells are? Because you're going to need to know for the exam. Anybody remember?
Like 70 to 90.
Yep. 70 to 95%. I'm gonna write it down one more time. Because if you remember, that's one of those few things that are not in your textbook that I want you to know. So yeah, according to the old textbook, your cells are 70 to 95% water. It's a lot of water. So what about the rest of it? Well, the rest of the cell consists mainly of carbon-based molecules. We've already talked a lot about carbon. And there's a reason for that. Carbon forms large, complex, and diverse molecules that are necessary for life. You don't need to write that down. It's just a preview of what we're about to talk about. So I met somebody, or emailed with somebody, I don't remember which, we were talking about drawing the molecules from lab 2. No, it was a meeting. I met with somebody, and I was explaining like the best way to do this, in my opinion, is to draw out all the carbons, like a long string of carbons, and then just put everything around that. It's all about, it should be the central, carbon should be central. Because carbon, that's right, that's what the person asked. They said, so basically when I draw these, I should put the one that has the most bonds in the middle. I was like, yeah, that's a great way of putting it. But in this case, the one atom that has the most bonds is carbon. Right? Carbon can bond 4 times, which I'm going to have on one of the upcoming slides. But because of that, it forms all these large, complex, diverse molecules. So anyway, these organic compounds are something called macromolecules. I'm not going to ask you any questions about that. I just want to say that throughout this chapter, I might use the term macromolecules or biomolecules. I might switch them back and forth. They're basically the same thing as far as we're concerned. But anyway, any questions about this slide before we move to the next one? We're going to talk a little bit more about carbon. All right, the next word for attendance is FDR, for the president. Today's his birthday. For that president, Franklin Delano Roosevelt. Any questions? All right, so like I said, carbon is versatile. It can make 4 covalent bonds. I think I've said before, you don't necessarily need to know how many types of bonds, but probably by the time you're done with this lecture and done with lab 2, you're probably going to be very familiar with the fact that carbon bonds 4 times. So yeah. Oh yeah, speaking of which, this reminds me of the meeting I just had with that person. Not only can it bond 4 times or make 4 covalent bonds, for the sake of your lab, let's say it has to make 4 covalent bonds. Because that was one of the things the person I was communicating with was a little bit confused about. So I just said, instead of saying carbon can, like, oh yeah, it can make up to 4 bonds. Let's just say it has to make 4 bonds, right? When you're drawing a carbon in those molecules, that carbon has to make 4 bonds. Whether that's 4 single bonds, 2 double bonds, or a single bond, or 2 single bonds and a double bond, whatever it may be, it has to have 4 bonds. Likewise, again, I'm just giving you some information for the lab that you have to do. Likewise, nitrogen has to have 3 bonds. Oxygen has to have 2 bonds. And hydrogen has to have 1. All that's in writing, so it's not like you need to take notes. I'm not giving you any new information. I just want to clarify that they have to have that many bonds. It's not like they can have up to that many bonds. As far as we're concerned, they have to. Again, 100 of course, there are exceptions to the rules, but this is how we're presenting it. So because of the fact that it can make 4 covalent bonds, which is more than any of the other big 4, remember the big 4 elements? It's more than any the other can make, so obviously it's going to be like the backbone of so many different molecules. You don't need to write this down either, but because of all this, because of the fact that it could have 4 bonds, there's an endless diversity of carbon skeletons that can vary in the size and the branching pattern. And this will be very important. Now, before I move forward, there's going to be a theme that you're going to see throughout this semester when we're talking about biology, this relationship between structure and function. You can tell a lot about how something will function based on its structure. And also, as we're going to talk about later in the semester, when you're comparing, for example, an extinct organism and a related organism that's not extinct, you can compare their structure and function and see how they're similar but different. And therefore, their structures are similar but different. Therefore, their functions would be similar but different. That'll come later. But again, that's a theme that we're going to talk about a lot this semester: structure and function. So anyway, let's talk a little bit about this. This is an example from the old textbook. These are just examples. There's nothing to write down, nothing to memorize. And it's only 4 examples, and these are also really simple examples. Like these molecules that are in our cells, a lot of them are huge and complicated. These are relatively simple molecules. So here you can see here the short little molecule. It's just 2 carbons, nice little shape like that. This one's got a look, to me, a phallic shape, to be honest. But anyway, you've got, just like here, you have a string of carbons, but then you have another a branch of carbons. Here, you've got like a longer version of this, but now it's a little bit extended with this double bond. And now you can see we don't have as many hydrogens as you do here. Then here, obviously, we have a ring. And how many of you are chemists? None, right? None of you guys are chemists. But that's okay. Even without being a chemist, we can look at these and see that they have different shapes. And a lot of times in chemistry, especially in biology, it really is like a puzzle, or like, puzzle pieces come together, or a key and a lock. So even without understanding what these molecules are or how they function, you can just tell they have different shapes. Therefore, they're going to fit into different things for that reason. And later in this chapter, you're going to learn a little bit about the chemistry of it. Like the fact that we have a double bond here, that means that's a lot of electrons right there. Again, that'll be important later, but we're not going to dig that deep into it. The point is, again, this relationship between structure and function. Even without being a chemist, you can look at these and realize they have different shapes. They're going to fit into different keys, different puzzle pieces, so to speak. So any questions about that slide? All right, carbon can bond with a lot of other elements. There's nothing to write down here. This is more of a refresher, a reminder, because this was previously in the chapter. But yeah, carbon most commonly bonds with hydrogen, oxygen, and nitrogen. Again, nothing to write down there. That's just a reminder of what the big 4 are. So remember when we first talked about the 25 elements that are in your body, we said these are the big 4 that make up about 96%. And then we talked about the medium 7 and the small 14. And I said of those, the only thing you need to know is the big 4, which is, again, carbon, hydrogen, oxygen, nitrogen. And if you've already done that too, you're probably already sick. Carbon, hydrogen, oxygen, and nitrogen. Maybe not so much nitrogen. It wasn't so bad in the lab. If I remember correctly, there was only an extra credit question of that one. So, any questions about this? All right, this is just an example. Once again, nothing to write down here. And I've already given this example, I think, in the last lecture, right? I tried to draw it. But methane is a great example. It's like the most basic carbon molecule. It's 1 carbon, 4 hydrogens, right? It's one of the most simplest organic compounds. It's abundant in natural gas. It's produced by prokaryotes, which as far as we're concerned is bacteria. So if you ever go to a swamp and it smells bad, that's what it is, it's swamp gas. Also cows, right? Cows cannot digest grass, just like we cannot digest grass. But they have bacteria in their gut. They can, so they can eat it, and it gets digested by the bacteria, and that's where the farts come from. Anyway, this is just the example from the textbook, so I wanted to include it. Again, I'm not going to ask any questions about methane. The most important thing about methane, in my opinion, is when I drew it on the other slide, I think in the last lecture, when I was saying, all right, carbon has to have 4 bonds, right? And I gave this one example of a carbon bonded to 4 other atoms. And I gave one example of carbon bonded to 3 other atoms. And then carbon bonded to 2 other atoms. So that was the point there, was just to remind you that I'm saying when I say something has to bond 4 times, I don't mean it has to have 4 different things bonded to it. I mean it has to have 4 bonds, whether it be, again, 4 single bonds, 2 double bonds, whatever. Also, for extra credit, I should have mentioned this in the last lecture if you want to look into it. There are some rare exceptions where carbon can bond to only 1 other atom. If you want to look into that for extra credit, feel free. But anyway, there's methane. No questions about that. So let's talk about some larger organic compounds. These are and this is great. I'm not necessarily going to ask about this, but this is a great comparison. This is from the old textbook. But there's some large organic compounds, some of which are the main molecules in gasoline. But meanwhile, similar organic compounds are also the fuels in your body, right? The food that you eat. And there's some similarities there. So for example, this is what octane looks like. That's what runs your gasoline engine, if you have one. And this is what fat looks like, dietary fat. So like that's got a lot of saturated fat or unsaturated fat. That's what we're talking about, dietary fat. And again, you guys might not be chemists, but you can look at this and look at that and be like, wow, that's similar. that might have a similar function. It doesn't always, but in this case it does. And we'll talk about it more later in greater detail, but right now I'll just go ahead and tell you the way it works, like I hinted at earlier when we were talking about chemistry and the energy it takes to build a molecule. Well, it took a lot of energy to build that molecule. It's a pretty long molecule. So when you break it, it releases energy, right? And that's what runs your car. And these things, right? That's three of those things, right? When you break them down, they release energy. And it runs your body. But again, those are just examples. You don't need to know that octane and dietary fat have those similarities. Are there any questions about that slide? The next slide I want to show you is more of a drawing that you might be more familiar with given lab 2. So we're still comparing octane to dietary fat. There's octane up there, and this is a saturated fat. Now granted, the last picture you saw, it's saturated fat. This is going to be a horrible drawing of it, but it basically looks like this. A saturated fat has 3 of them. You don't need to write that down. We'll talk about that later. I know that looks like an E, but that's supposed to be 1, 2, 3 of these things. But again, similar structure, similar function, right? When you break down either one of these molecules, you're going to release energy. And that's something we're going to talk about, I think, in the next chapter. We'll hint at it here, but it's the next, the material for exam 2, where we really start talking about energy. But right now, we're just building you up to it. So again, as far as the exam is concerned, this is not important. So are there any questions about it? All right, here's some more information that's not in your textbook, and I'm not going to test you on it, but it's good to know. So when we move forward, you have a greater understanding of what we're saying. So we're talking about all these different carbon molecules with all their different shapes and sizes. Their unique properties depend very often, not just on the carbon skeleton, which we talked about. Remember, like I said, it's all about the shape. Well, not all about the shape. It's a lot about the shape, right? Where this thing can only fit in that thing, and this thing's too big to fit in that thing. That's part of it. Another thing is the atoms attached to the skeletons. And those things are called functional groups. And again, I don't necessarily think I'm going to test you on that word, and this is not in your textbook. But I think you need to know what functional groups are, because we're going to talk about them this chapter, and then in later chapters too. So I think it's important to know what they are. A functional group is a group of atoms directly involved in a chemical reaction that's attached to a chemical or a carbon skeleton. So in other words, like you might have this basic carbon skeleton, and it might not be much to it chemically. These things that are attached to it might make it, polar versus nonpolar. Remember we talked about that, polar versus nonpolar. Polar is where there's an unequal distribution of charge, and nonpolar is it's equal distribution of charge, things like that. I'll give you some examples on the next slide. Each functional group plays a role in chemical reactions, and many biological molecules have 2 or more of them. Once again, there's nothing here that's going to be on the exam. I just want you to understand the general concept of a functional group. That reason on the next slide, I've got a chart that's also not from your textbook. It gives a bunch of different categories of functional groups, and we're really quickly going to talk about them, but you don't need to know anything that's on the next slide either. It's just a good background information. So when we start talking about some really complicated molecules later in the semester, and even though we don't dig into it for this 100 level class and talk about why they're so complicated, you just have this knowledge in the back of your head to understand the basics of it. Now, before we do that, is your next word for attendance. I'll give you some time to write that down. Richard Theodore Greener for extra credit on a separate email. Well, no, this one's, on a separate email, so, doesn't matter when you send it. You can look up who that is. I think it's their birthday today. I don't remember if it's their birthday, or the day they died, or the day they did the thing that they're famous for, but probably not the day they did the thing they're famous for, since it's January. Anyway, that's the next word for attendance. Any questions about this slide before I give you the chart, the table that shows some examples of different functional groups? All right, so here's some different functional groups. And once again, I love this table because even though you don't need to know any of this, these are words I'm going to be using later in the semester. You're going to hear me I might say hydroxyl every once in a while. I might talk about methyl groups. I might talk about carbonyl or carboxyl. Definitely gonna talk about amino groups later. And definitely gonna talk about phosphate groups later. Not that you need to memorize any of this, but now you've been exposed to those words that we're gonna use later. And look at these properties, right? This one's polar, this is nonpolar. And why that matters is if you had two different molecules with a carbon skeleton and one of them had this on it, one of them had that on it. What I haven't told you yet is polar versus nonpolar, they repel each other. So if you have those two molecules, they're not going to come together. At least based on the very simple way that I'm describing it. There's other factors to think about, but anyway, generally speaking, polar things and nonpolar things don't go together. So again, this R, that just stands for, I don't know why they chose R, but R just stands for, basically as far as we're concerned, carbon skeleton, right? So this means this thing here is attached to some other carbon skeleton that starts right there, starts right there, so on and so forth. What else? Another thing to think about, again, charged versus uncharged, right? This thing, in this example, carboxyls, charged, right? It has a charge. It ionizes. It releases hydrogen ions, which means in a sense, as far as we're concerned, it's acidic, right? And we know acidic and basic things kind of neutralize each other. So again, without even being a chemist, you can just understand that these functional groups, based on their size and their shape and their properties, they just do different things. Anyway, any questions about that side? Okay. On a molecular scale, many of these biological molecules that we're going to learn about are gigantic. That gives them the name macromolecules. So you don't need to write this down either, because, as I mentioned earlier, there's 4 categories of molecules that we're going to talk about. Excuse me. Technically, only 3 of those categories are considered macromolecules. The other one is not. But I'm not going to ask you any questions about that. But anyway, the 3 categories that we're going to discuss individually, along with the other 1 category that's not listed here, is carbohydrates, proteins, and nucleic acids. And the one that's not listed here is fats, triglycerides. So yeah, we're going to talk about all 4 later. And just so you know, before we even jump into it, I'll go ahead and tell you now, so you can think about this as I'm talking about it. For the exam, a lot of what the questions are going to be about is the monomer versus the polymer. And I'll tell you what that means. Right? A macromolecule is a polymer, and a polymer is something that's made by stringing together smaller molecules, like repeating molecules. And those repeating smaller molecules are called monomers. Like this little picture here, I know it's a little bit goofy, but that's a monomer, 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 and all together they come together to make a polymer. And you can see that they're pretty much identical, right? It's like just repeating the same thing makes the polymer. But anyway, what I'm getting at here is for this exam, a lot of the questions are going to be, I'm going to describe something by its polymer. I'm like, all right, this thing, here's the polymer. Now you tell me what the monomer is. Or it might be vice versa. Right? I say, here's the monomer. And you tell me, knowing that that's the monomer, is it a carbohydrate? Is it a protein? Is it a nucleic acid? A lot of that, the exam is going to be that, at least that portion of the exam. When we're talking about the macromolecules, the biological molecules, it's going to be a lot of questions where you differentiate, and that's going to be the way you differentiate. One of the ways, one of the big ways. Here's a new term you need to learn and know: dehydration reaction. So these are the things that basically build polymers, and we're going to be talking about dehydration reactions throughout, well, not throughout, but for a lot of the semester. Dehydration reactions link the monomers together. So again, you can see these in this goofy picture, these things are holding hands. And as you can imagine, those things are basically covalent bonds, even though it's a silly picture. They're little arms holding each other, those are covalent bonds. It is a dehydration reaction that brings them together. I'm not going to ask you this, but this will be handy, the second bullet point. Dehydration reactions remove a molecule of water. That'll make more sense in the next slide. And again, that's not really important as far as we're concerned. I'm just hoping it helps you, because this, in my opinion, is easier to remember. The dehydration reaction, nice and easy. It brings together polymers. But later, I'm going to tell you about another reaction that's called a hydrolysis reaction. And when I was a student, those two things sometimes I would get them confused in a quiz or exam because they sound so similar. Dehydration, hydrolysis, they both sound like they have something to do with water, and they both do have something to do with water. So anyway, on the next slide, like I said, this should make more sense. But before I go to that next slide, are there any questions about this? Yeah. Really quickly to point out too, if you're following along in the textbook, I'm doing this in the same order that the old textbook did it. So it is a little bit out of order, but it's the same information. Anyway, here's a dehydration reaction. We're bringing together things. That's a better way of putting it. When we're bringing things together in this course, we're bringing together two molecules, two or more molecules. We're calling that a dehydration reaction. And you can see this is how it happens. It's not going to get this specific in the exam, but you can see this one has an OH off of it. This one has a hydrogen. Obviously, two hydrogens and an oxygen make water, right? So those come together and you make some water. But for the exam, the way this is important, if you see any, I'll give you some chemical reaction, and it'll be like, I don't know, X plus Y. I mean, it's not going to be X plus Y. It'll be some molecules that you haven't heard of, and maybe some atoms, or maybe some words I just make up. Anyway, X plus Y gives you XY and H2O. If you see the water on the right-hand side of the reaction, you know it's a dehydration reaction, because you have dehydrated the reactants, right? Yeah, nice and simple, right? Dehydration reaction, you've dehydrated the reactants. You've produced water in the products. And really quickly, just to remind you, since I've drawn a chemical formula, I want to remind you that in real chemistry, a lot of the times, you'll have a double arrow, right? Because a lot of times chemical reactions can go both ways. Actually, this is a great example. Even this thing, whatever this example is, we have this 1, 2, 3, 4, 5. There's 5 monomers to make this polymer. They came together in a dehydration reaction. Meanwhile, and we're not showing it now, we'll see it later. Meanwhile, you could take a water molecule and break that molecule up. So yes, this chemical reaction is reversible, and this hypothetical chemical reaction is reversible too. But for the sake of simplicity, for the exam, whenever you see a chemical reaction, it's always going to be reactants on the left, products on the right. The arrow will always be pointing away from the reactants to the products. And again, if it produces water, it's a dehydration reaction. So it's almost like, think about like you. It's because that can sound a little bit confusing. But if you become dehydrated, it's because you produced water, right? You've sweat, you've used the water up in your body, you've urinated it, whatever, but you've produced water, right? Therefore, you were dehydrated. So that, again, I don't want you to get confused. It's kind of hard when you think about, like, we produced water. How is that dehydration? But again, think about your bodies. When you produce water, you are becoming dehydrated. Anyway, any questions about that? All right, like I said, the opposite of that is called hydrolysis. Hydrolysis is when we're breaking the molecules. Obviously, that's going to happen in things like digestion. Break down molecules, which is important. You can almost think of it like Legos. Like if someone has a set of Legos and it's built a house, but you need those Legos to build a car. You need the Legos. You need those individual pieces. You don't need the house. So you break down that house of Legos into its individual pieces, and then you put it back together to make what you wanted to make, which was a car. That's a hypothetical example. Same thing here. A lot of the stuff you eat, your body doesn't necessarily need it in that form, so it breaks it down to its building blocks so it can build other stuff with it. At least that's how we're focusing on it in this chapter. Later, as you're going to learn, not only are we breaking it down so we can have those building blocks so we can build other things, but also, as I've hinted at previously, when you break those things down, you're releasing however much energy it took to build them. And obviously your bodies need energy too, right? We don't just need stuff to build, we need energy to build. And again, a hydrolysis reaction is the opposite of a dehydration reaction. I have no good way of helping you remember hydrolysis breaks apart stuff, right? I have no good ways of helping you remember that. I'll draw this again. XY plus H2O, to use the same chemical formula I used last time. With the same hypothetical, gives you X plus Y, right? I have no good way of helping you remember that. Again, dehydration, the way I told you to remember that is because you produce water when you become dehydrated. That's how you become dehydrated, is because you're producing water. So just like that, in chemical reactions, when you produce water, it's dehydration. I don't have a good way of helping you remember that. I guess just by process of elimination, if it's not dehydration, it's hydrolysis. Anyway, any questions about those two concepts? All right, we're making good time. We have catching up to do. All right, so there's no questions about that. We'll go kind of step back again, look at the big picture. These are the 4 biological molecules we're going to talk about. Again, you don't need to write them down if you don't want to right now, because we're going to talk about each one of them separately. Carbohydrates, lipids, proteins, nucleic acids. You need to know the difference between the 4 of them on the exam. Like you're going to need to know, for example, and you don't need to write this down yet because I'm going to say it again later. For example, carbohydrates are hydrophilic, and I'll explain that later. Lipids are hydrophobic. You need to know that amino acids are the monomers of proteins, while nucleic acids are the monomers, well, nucleic acids. So anyway, again, it's going to be a lot of comparing and contrasting on the exam. So keep that in mind as I'm teaching you this stuff. Also, to let you know, not only do we need to know the difference between, carbs versus proteins or whatever, between the 4 categories, then within each category, we break them down. So you're going to learn about different types of carbohydrates, and you're going to need to know the difference between them. You're going to learn about different types of lipids. You're going to need to know the difference between them. I'm going to introduce you to different types of protein, but honestly, you don't need to know the difference between them. We'll talk about those later in the semester. Nucleic acid, same thing. There's basically 2 types of nucleic acids. We'll talk about it, but later in the semester is when we really dig into that. So I guess basically it's these 2, and you're really going to need them. We're going to break down some more. Anyway, these pictures, I chose these pictures. I think it's interesting because what we're saying is, of all living things, right, whether it's a human like us, or the bacteria that's inside of us, or the little bugs that live inside your skin pores in your face, they all have the same, all their cells, we all have these same 4 biological molecules, which is what I think is interesting. And these pictures, anyway, just represent, this is probably one of the smallest animals. Does anybody know what that is? Not that you need to, I'm just curious. That's a tardigrade. They're really interesting. You can radiate them, you can put them in space, you can dehydrate them, they're really hard to kill. They're also really tiny. And then this happens to be the largest, well, people argue about this, but some people say this is the largest organism on Earth. You're probably looking at it and like, well, you're an idiot because that's a bunch of trees. It is a bunch of trees, but it's all one organism, oddly enough. And I'm not saying, like, if you look out the window, all those trees are one organism. I'm saying this particular stand of trees happens to be one organism. So that's unimportant. If you're interested, you can look any of that up for extra credit. So anyway, any questions about this slide before we jump into our first category, which is carbohydrates? All right. So if we were not in biology class, and you're just talking with somebody, or reading something on the internet or whatever, when someone says carbohydrate, what do you think of? What does that word mean to you? And again, I don't mean in a biology sense, unless you just happen to think biologically. What do you think most people are talking about when they talk about carbohydrates?
Calories.
Calories, that's good. Which is funny you should mention that, because we will talk a little bit about calories now and a little bit more for exam 2. But yeah, calories. And to point out, carbohydrates and the other things too also provide calories, but okay. What else? What if I say I'm cutting back on my carbs? What does that mean? Because, to her point, I mean, if I cut back on my carbs, I will be cutting back on calories, but it's a little bit more specific than that, right? If I cut back on my carbs and I just eat nothing but steak, I'm still getting plenty of calories, right? So what else? What else do you guys think of? When I say I'm cutting back on carbs, what does that mean? What am I talking about? So I'll wait a little bit. Because like I said at the beginning of the semester, I don't want to be sitting up here talking to you. I'd rather be talking with you. What if I say I know this looks funny right now because I've gained a ton of weight, but what if I said, I'm gonna run a half marathon tomorrow, so tonight I'm gonna carbo load? What am I talking about? No one wants to guess? You just too shy? No. Yeah, carbs, good. To you, what does that mean? Yeah, but he's right, right? When I say carbo load, I mean eat a bunch of carbs, get energy. That's exactly it. What I eat if I was saying I'm gonna carbo load. Don't be shy. You guys know this. I know you know this.
Stuff.
There you go. Pastas, stuff like that. Good. So yeah, like this picture. I think this is from the old textbook. This is a great picture. I love it because here we have spaghetti, and I think that's what most people think of when they think of carbs, even though you guys are hesitant to say carb. Pastas, bread, rice, that kind of stuff. But it also includes the sugar that's in Coca-Cola. That's also, or any soda. I don't know how much the old textbook got paid to include Coca-Cola, but anyway, yeah, that's also carbs. And that's what I kind of wanted to get at. So all the things that you think about of carbs, that's probably true. But there's also other stuff that you might not think of as carbs, and that's the point I was getting at. The carbohydrates include sugars and polymers of sugars, and you should know that. Of course, I'm going to give you a different word other than sugars, so I guess even though you should know that, I don't know if you want to write it down, whatever. Just for the exam, I'm not going to use the word sugars. I'm going to have another term for you. But yeah, so a sugar is a pretty basic molecule. And when you put a bunch of them together, you get some of these more complex molecules, right? So like you got the pretty basic molecule of sugar in that soda. And if you were to take those same molecules and string them together, you would get the starch that is in that pasta. That's all I was getting at there. And this is just an introduction. We're going to talk a little bit more about it, obviously, in the next few slides. Are there any questions so far? All right, and really quickly, this is not a nutrition class, so even though I'm going to be talking a lot about nutrition, because that's what how you can relate it to your everyday life. When I said you're going to need to know the differences between this type of carbohydrate and that carbohydrate and this biomolecule and that biomolecule, these will never be in any of the examples. So don't worry about that. Again, I'm just using them as examples because this is what you're familiar with in your everyday life. You also probably don't even need to write this down yet either, and I'll tell you why in a second. But in animals, carbohydrates are the primary source of dietary energy. This is why I like what Nika said, because when I said, what do you think of? She said calories. I mentioned, well, everything has calories, but again, it's the primary source. As a matter of fact, there's one carbohydrate that we're going to learn about that's really important that we're going to talk about in an exam too, a lot. It's called glucose. It is our primary source of energy. I mean, obviously we eat stuff other than glucose, but that's our primary source. It is also the raw material in animals for building other molecules. So again, like I said earlier with those hydrolysis reactions, you eat these big molecules, your cells break them down to the building blocks so they can rebuild it into the things that we need. In plants, I don't know, this is from the old textbook too. I don't like this now that I think about it. In plants, they wrote that it's the building material for much of the plant body, which is true, which is very similar to that bullet point. But also, it is also, I don't say dietary energy, but carbs are also the energy source for plants too. And that actually will be written down in the slide later. So I don't know, in a sense that I'm thinking about, it's kind of a dumb slide. I don't remember if it's from the old textbook or the old one, but it's kind of the same. Now, of course, you guys know plants go through photosynthesis, which we'll talk about later. And they get their own energy from the sun. But guess what? They're only using that energy to make carbs. And then they do what we do, the same way we do it. They break down the carbs, and then they use that energy to live. So, and again, we'll talk about that later, but there's your first preview in case you didn't know. I think, again, most people know that plants get their energy from the sun, but they don't use it to live. They use it to make carbs, and then they use that energy to live, just like we do. Anyway, any questions so far? Only got 5 minutes. Let's at least finish. Again, there's different categories of carbs. I'm going to talk about them. And like I said, of all the 4 biological molecules, well, 3 of them, carbs, proteins, and nucleic acids, you're going to need to know the difference between, you need to know their monomers and their polymers, right? So let's jump into the monomers for carbohydrates. They're called monosaccharides. So that will be important for the exam. You need to know that monosaccharides are the monomers of carbohydrates. Even though it's not on the slide yet, this is one of the easy ones. if the monomer of a carbohydrate is called a monosaccharide, does anybody want to guess what the polymer of a carbohydrate is called? Don't overthink it. The monomer is called, the monomer of the carbohydrate is called a monosaccharide. Anybody want to guess what the polymer is called?
Polysaccharide.
Yes, polysaccharide. Unfortunately, they're not that simple, as you're going to learn when we get to proteins, for example. But anyway, yeah, monomers are the monosaccharides are the monomers of carbohydrates. What that means is they cannot be broken down. A monosaccharide cannot be broken down into smaller sugars. You can break it down, as you're going to learn when we talk about respiration. We break down monosaccharides to release that energy. But once you break it down, it is no longer a sugar. You cannot make it any smaller without, it would be no longer a sugar if you break it down. One of the things that's not going to matter for the exam, but I'm going to go ahead and tell you, these two bullet points. I really don't think I'm going to ask you any questions about this. But when we're talking about them, they usually end with the suffix -ose, like triose, or pentose, or hexose. So when we're talking about sugars, yeah. If a sugar has 3 carbons, we call it a triose. If it has 5 carbons, we call it a pentose. If it has 6 carbons, like the ones we're going to talk about a lot in this semester, they call it hexose. So again, I'm not going to ask you about that on the exam. And of course, even that will be broken down. So for example, on the next slide, let me give you an example of 2 different hexoses. And there's a bunch more than that too. So again, you don't need to know this for the exam. And also just to let you know, these are also categories. So there's all kinds of sugars that fall under this category. All kinds of sugars that fall under this category. All kinds of sugar that fall under this category. And they're all monosaccharides. Some common examples. Again, this is not a nutrition class, so I'm not going to ask you this in an exam. But you have glucose that's in like Gatorade, fructose that you can find in fruit. And it turns out you can find both of them in honey. That'll be the picture I show you on the next slide. So again, this is not a nutrition class. I'm not going to require you to know any of the examples, but I will say this. Some of them will be sprinkled in, not because you need to know them, but just as a hint. Like, all right, I'm going to tell you the things you need to know about a monosaccharide, and maybe I'll throw in the hint, like to help jog your memory that, hey, it's also in Gatorade. This is an example. So any questions about this slide before I show you some sugars that are in honey? All right, so here's glucose on the left and fructose on the right. You can find them both in honey. You do not need to know that, but one of the things you do need to know is this new term that I have not shown you before, and it is isomer. An isomer is when something has the same formula in different arrangements. So for example, if you were all to work together as a class, where you had to draw those molecules, I bet if you all looked, that's the last question, that's the last one you had to draw, was C6H12O6. And off the top of my head, each one of you drew it in a different way. Well, there wasn't that many to turn it in yet. But anyway, you can see here, this is the same chemical formula, but it's just drawn a different way. So that is what an isomer is. And like I said earlier, with this whole relationship between structure and function, you can see these two have very similar structures, only slightly different structures, right? And it just so happens in this case, their function, if you will, is very similar but slightly different. Matter of fact, glucose and fructose, they're both sweet. Fructose just happens to be a little bit sweeter. Just because we have this instead of this. And that just makes it a little bit sweeter. It's interesting how these things work. But again, you don't need to know the difference between glucose and fructose. You just need to know what an isomer is. An isomer, again, has the same formula with different arrangements. And it's 8:50. So when we get back, we'll finish our discussion about isomers on Monday. And don't forget to send me the email. If you're watching the video, include the screenshots and the attendance words, and then one question about the material. And if you're live, you don't need to do the screenshots, just the words and the letters. That's it. I'll be online and in my office for office hours. Hope you guys have a great weekend. You only came in late, right? Nope, everybody's accounted for. Is it? Maybe, I don't know. There was definitely a few people I couldn't see the screenshots yet. Oh, in the lab? Pre that, okay. Yeah, let me, let's go ahead and take a look now. Really quickly. So, sorting.